Ça fait plaisir d'accueillir aujourd'hui Dr. Gazda, I use your short pour une conférence. Gazda est arrivé au Québec récemment. Elle a commencé un poste comme professeur adjoint au département de sciences biologiques de l'Université de Montréal en octobre 2023, donc ça fait très peu de temps. Et avant de se joindre à l'Université de Montréal, elle a fait ses études, une partie de ses études en Pologne, à Cracovie pour sa maîtrise et son doctorat, son baccalauréat. Puis ensuite, elle a fait un doctorat au Portugal, où elle a commencé à travailler sur la génomique fonctionnelle ou l'architecture des traits complexes chez les oiseaux. Puis un de ses, si je ne me trompe pas, un de ses papiers, ou son papier de doctorat, a fait le cover de la revue Science sur les bases génétiques du dimorphisme de couleur chez les donc dimorphisme sexuel de coloration. Ensuite, elle a fait un postdoc avec Camille Berthelot, qui est dans l'Institut Pasteur à Paris. Ceux qui travaillent en génomique des poissons peuvent peut-être entendu parler des travaux de ce labo-là. Elle fait des travaux sur la génomique comparative des poissons. Mais ce qui intéresse toujours Gazda, c'est les oiseaux. Et donc maintenant, on va nous parler de ces travaux récents. Thank you so much for
Merci beaucoup, Chris. Oh, do you hear me well? Awesome. So thank you so much for this kind introduction. So today I will talk about looking for the genetic basis for carotenoid coloration in birds, but it also turns out that not only in birds, we will get there. And it's mostly work I've done during my PhD, also some work I've done after, and I'm trying to come back to work on canaries in my lab. But since I started in October and I'm trying to get birds and so on, I don't yet have results, so I'm gonna show you the previous work. So first of all, I really like to work on birds because they enable to study a lot of interesting phenotypes, interesting traits. Of course, is a great model to study many things, but I have been thinking, so well, coloration is one thing, but the other thing, I'm really into it. It's song, and then working on any other model species than birds, it's very challenging. So I think, in particularly for song, the birds are a very good model, but there's of course a lot of other traits. So I had a project on the flight and athletic performance in pigeon, and then as I said, I studied a lot about coloration, and in particular about carotenoid coloration. So you can look across different species trying to do the comparative genomic study in two different ways. So you can find the trait polymorphism, like we have here, example of different blue butterflies, so across different species, or you can look within one species, and that's particular case of domesticated animals such as dogs, but also the canaries. And then within one species, we have many different phenotypes that we can try to study. And canary birds are particularly good model for this kind of work because there has been a recent strong selection on different phenotypes. So it's basically the canary breeders that did around 400 years, well, depending what kind of trait, but in general, around 400 years of experimental evolution trying to select for particular phenotypes. And probably not everyone realized, but there's over 200 breeds. So I will talk about colors, so we will touch a lot different colors, but they have also different type of body, posture, feather structure, and there are also the song breeds that I mentioned that are a good model to study the vocal learning and so on. For many of them, we also know the patterns of the inheritance because the breeders are very excited about them and created a lot of resources. So we usually know if the trait is dominant, well, if the gene is dominant or recessive. So that's also very useful. And recently there has been a lot of resources created for canaries. So when I started my PhD, there was a reference genome, but then we decided to improve it. So we published another version of the canary reference genome. And then I think like last year or 2 years ago, there was even newer, more improved reference genome of canary with like long reads, and it's almost normal level assembly, which is very nice, and it helps to have better quality data and better idea, especially if the trait and the architecture of the trait, it's not that obvious, but I will get there. So I told you it's over 200 breeds of canaries. You can see examples of them here. So as I said, a lot of coloration. So the coloration, yellow, red, and orange, it's related to carotenoids. But of course, we have a lot of like brownish phenotypes, and that's related to the melanin. And melanin, I haven't studied. I focused on coloration, on carotenoids, and what I told you about the weird structure of the body and the feathers. Here we have the frisado canaries and also Italian gibberlicus that looks like a little bit anorectic canary, and they are very interesting, but sometimes that was such a strong selection that they also tend to have some health problems and health deficiencies. And sometimes it's challenging to keep them because a lot of work was done by the breeders, including some like hair of the cheeks. So the canaries can destroy own eggs, and then they really need. So it was maybe the selection was pushed too far, and like a lot of those phenotypes would never exist in nature for those reasons. Coming back to the carotenoids. Carotenoids are very important players in the bird's physiology. The birds cannot produce them themselves. They have to acquire them from the diet. So it's the plants producing carotenoids, and then the birds eat, for example, seeds. They are important for immune response and antioxidation. And interestingly, it's not good not to have carotenoids, but actually it's not good to have too much carotenoid. It can be toxic. There has been some studies showing that too much carotenoids is also not good to birds, and we will get there too. And then they impact the whole all the physiological processes and metabolism. And of course, they can create nice ornamentation. So the bird can have all the feathers colored with the carotenoids. It can be also ornamental coloration. So for example, only beak and the bird parts. Yeah. So what I'm interested in is I try to integrate genetics, ecology, and evolution and understand how the traits evolve, how adaptation arise by linking the genotype with phenotype, and then having this link, understanding what gene is behind the trait. Well, here I will be talking about the simple traits, so 1 or 2 genes, not complex traits that are having hundreds of genes interacting. And then this allows me to learn a lot about biology. So for this kind of studies, first of all, it's important to choose a good model. So here I have example of the red and orange morph of parrotfish. So it's one species, and then they have 2 morphs. Then, of course, I need blood samples to sequence the DNA and compare compared those tumors across the genome, and using different populations statistics. Here it's the genetic differentiation, FST. You can find the region differentiated in the genome. Of course, many times it's much more complicated than that. This is like the best Manhattan plot I had, but sometimes, for example, if the Manhattan plot is messy, it's useful to know if it's sex-linked trait or autosomal. That helps to look in the right place for the candidate genes. And then, of course, well, knowing the gene name, it's cool, but then it's also nice to look at the patterns of the gene expression in related tissues or cell culture. And then finally, I think the coolest what you can try to do is to find the link between those genes and other biological or physiological features. So here, for example, those birds, there's the mutation in the, well, I will get there, but like in the gene that enhances carotenoid coloration. And that leads to the fact that in the eyes, in retina, they don't have the red cones. So they have others, but not red. So therefore they see differently. And I think this is very interesting and very cool. So first of all, talking about the carotenoid coloration, the first question was, how do birds acquire carotenoid coloration? So good model to study that is the white recessive canary that doesn't deposit any carotenoids, and then just compared with the regular yellow canary, but it could be also, well, the yellow was the best because the red I will get later. So what we did, we sequenced the whole genomes in the pools. So there was a pool of white recessive canaries and the pool of yellow canaries, and compared across the genome. So you can see here that the plot is a lot of less clean than the first one I showed, but you still can see a peak here in chromosome 15, the strongest signal in the genome. And here we had the individuals from different breeds. That also helps because so of course they have the coloration, but then they have also many other traits. So the comparison of the specific breeds can be very messy. So that including more breeds that have or don't have the trait can be helpful to try to increase the signal. And also we have the wild birds from the Canary Islands that don't have those traits. And then, so whole genome sequencing, Illumina, 100 base pair reads. Well, this changed a lot across, recently, but that's from 2017. So that was what we did. And then, well, moderate coverage around 20x. So we found the signal in the genome, and then the peak in the region of where the peak was located. There's the SCARB1 gene, the scavenger receptor class B member 1. And there were some links already from studies in worms showing that this homolog of this gene was implicated in things related to carotenoids, so that was a good hint. But then to confirm exactly what is going on in those canaries, we found the isoforms of the SCARB1 from different tissues. And basically what happens in the white recessive canaries, then there's the splice site mutation that leads to the skipping of exon. It's not the only isoform. So you can see here in yellow, there's only the wild type isoform that is the normal isoform of the SCARP1 gene. But then in the white recessive, well, there's different places that the splicing can occur. Therefore, there's like different isoforms also in different tissues. But the most abundant, it's the one that is related well, to exon 4, skipping exon 4. And in wild type, there's only the wild type isoform in the tissue. So therefore, with those cloning experiments, we were able to show that indeed the mutated SCARP1 leads to the lack of ability that the birds cannot deposit the carotenoid pigment in the feathers. And then we also did the cell culture experiment to check if indeed the function of SCARP1 is crucial for this metabolism of the carotenoids. And in the control and in the isoform 4, we saw like very little concentration of the carotenoids, which might be also related to the endogenous protein. And in the wild type isoform, we can see that the cells acquire a lot of carotenoids. So and it was done in the avian fibroblast cell lines. So with this, okay. So to sum up, I showed you that the SCARP1 it's an essential mediator of the expression of carotenoid-based coloration in birds. What I didn't tell you, but the SCARP1 is also important for the cholesterol metabolism. So in this way, we can link the processing of the lipids and cholesterol with the coloration. And since it was shown that it was important for the silkworms, it seems also to be quite ancient and conserved mechanism of carotenoid uptake in animals. So now we know what is the essential gene for the carotenoid deposition. But the second very important and interesting question is, when we look across the birds, quite often we can see which birds are males and which one are females. Well, there's some outliers from this rule, but in general, the shiny, colorful birds are males and the drab birds are females. And then there was a question, what is the gene regulating the sexual dichromatism, so those differences in the level of coloration in the birds. And yeah, it's well, widely spread across birds. There's also different types of pigments. So here it's not carotenoids, it's the structural coloration. But it's widely spread across phylogeny, and it's also the major driver. The sexual selection is probably major driver of those differences. But it's a little bit challenging to study it, because in nature, usually you have species that either have or don't have the sexual dichromatism. But again, coming back to the work that was done by the canary breeders. So that's actually byproduct of the selection process. So the idea was similar, like for the blue rose, the canary breeders were really dreaming of having the red canary. But unfortunately, the canaries with the carotenoids were So how to get to the red canary? They came up with the idea of crossing the common canary with the red cysteine. So you can see well, this is the male, and then they have a lot of red carotenoid. They have also some melanin. The hybrids didn't look super attractive, but with the process of many back crosses, in the end, they created two breeds. So I'm showing here the mosaic canary, so the canary with the sexual dichromatism. So it really looks like the hybrid between those two, with the carotenoid patches in the face, in the feathers, and in the tail, and much more carotenoids in the male than in the female. And the other breed was also the red canary, so the whole bird just red. So since this was the process of introgression, the signal coming from this gene, it's very strong. So yeah, basically the genome looks like the canary genome with the region of regions that were introgressed. So here you can see, so it's mosaic canaries, and there's actually two types of mosaics that we also included. So there's the red mosaic, and then there's the yellow mosaic. With the yellow canaries, and then we compared also mosaic with the red cysteine to check for the introgressed. And here we have both red. So first of all, there's a strong peak here, but you can also see a significant peak here. And then I told you that it's the introgression process and the signal is very strong. So we can see here two things. This region in the genome is responsible for the red carotenoid coloration, and I will come back to that a little bit later. We don't really see it here, well, a little bit, but not really, because here we compare the red individuals. So then this dilutes the signal, and then we can see the right small introgressed region responsible for creating the sexual dichromatism. So in this region, well, there were quite some gene located. Then we did the genotyping, and we restricted the region to PTS, TEX12, and BCO2. And BCO2 was a very good candidate because it was previously shown in other studies related to fat color, for example, that it's important for the carotenoid coloration. And then, well, to confirm indeed that like how the BCO2 works, and this is what helps with the sexual dichromatism. We did the experiment, so we had the birds in the lab, and then we plucked the feathers to see what are the genes expressed in the regenerating feather follicles, because well, once the birds have the color, then the gene doesn't necessarily have to be expressed. It's more at the moment when those colors are deposited, created in the feathers. So in the pigmented feather follicles, we saw, so here we can see the yellow, that's carotenoids, and here we can see the BCO2 expression. So when you have the pigmented feather follicles, there's not so much BCO2 expression, whereas in the unpigmented feather follicles, you can see that there's not really much yellow here in comparison with pigmented, and there's much more BCO2. So here you don't really see the violet, whereas in the unpigmented feather follicle, there's a lot of BCO2. Yeah, so the more BCO2 active, then the less carotenoids, which makes sense because the BCO2 is the gene that basically is responsible for removing the carotenoid. So they are processed, but then they are not deposited in the feathers. But we were also curious if this is only a case in canaries, or it's maybe more widespread mechanism for controlling sexual deformatism in birds. So we sampled the birds and collected the birds and then did similar type of sampling. So removing the feathers to have the regenerating feathers, and then to take the tissue samples, in this case it's skin, and then quantify the gene expression. And here we are in nature, so no more domesticated birds. We have the wild birds, so it's the common canary, the ones from the Canary Islands. And with the colors, I represent here the amount of the carotenoid difference. So here we can see that the males have a lot of carotenoids in the breast, and the females not so much. And then in the belly, the males and the females have the carotenoids. So there is some difference, but not too much. And then we look at common canary, European serine. That in European serine, there's much bigger difference between the male and female. And in house finch, where we have the red coloration and a lot of coloration in the breast, but not so much in the belly. So first of all, we quantified the differentially expressed genes. So in the European serine, we find a lot of differentially expressed genes between those regions. So here I'm comparing the breast of male with the breast of female, and belly of male with the belly of female. So we can see there's a lot of differential genes. And in the regions that there's not that big differences, there were less differentially expressed genes. But what are those genes? And also one more interesting thing comparison here is that we have the common canary and European serine, and those are the sister species. So we also compared the tissue patches across those two species to see differences in the sister species, but as expected, you see a lot of differentially expressed genes. So the smallest number was of differentially expressed genes shared was between patches, and then there were a lot of differentially expressed genes between species and quite some across sexes. But then the other question was like, what are those genes? And one of the top differentially expressed genes was BCO2. That was true in the European serine for literally every comparison we did. So between the sexes, between the patches, and also between the Canary and European serine for the specific comparisons. But that was not the case for the house finch. So the BCO2 was not differentially expressed there. So it seems that there's more genes regulating the sexual deformatism. So what we've learned so far is that the deformatism in birds is generated by a simple molecular mechanism driven by a large effect gene, that is BCO2. The function of BCO2 exerts in the peripheral tissue, so in this case, it's in the feathers. And it also regulates sexual deformatism in some wild birds, such as common canary and European siskin, but not all. And it seems that carotenoid signaling might be shaped more by cellular processes than resource limitations. So there's a lot of discussion here around honest signaling and males having to acquire a lot of carotenoid to show that they are attractive, beautiful males. But it seems to be a little bit more complex, because with BCO2, I'm showing that it's actually the BCO2 working females to remove the carotenoid instead of like increased activity in the maize. So that's probably a little bit complex than that, but it might be also related to those risk of BCO2 being potentially toxic to the tissue. So maybe that makes it more expensive for the maize to be able to process. One more thing about the BCO2. So at that time, well, we were curious about those traits, but then we were a little bit, well, I would say disappointed because every study, and well, okay, on birds here related to red carotenoids makes sense. It's BCO2 again, but also like in other studies in the lizards, also related to coloration, BCO2 was popping up all the time. Well, it seems to be an important gene for regulating the amount of carotenoids. So here it's another example. So those are well, first of all, the products of this process of introgression of the red coloration. But on top of that, that's a special breed of the canary. It's called urucum. So they have very strongly colored beak and legs. And this is one of the breeds that I told you that they have a lot of physiological problems. So the reproduction, so breeders like it because, well, it's beautiful bird, but it has a lot of health issues. So it's not easy to well, first of all, reproduce them, and then second of all, to take care of them. So that's probably also related to the thing I told you already, that BCO2, when it's too much, it can create problems. Well, so here again, we have the Manhattan plot comparing the Urucum breed with other breeds, and we see the strongest signal again in the BCO2 region. So in this case, there is a missense mutation in BCO2 gene that results in the loss of function of BCO2. So that's why there's like so much carotenoids in all the possible tissue, because the birds don't process them. So yeah, that mutation, it's not present in any other bird species. It's specific to the Urucum canaries. And it seems that well, this specific mutation causes the structural distortion of the protein, and that leads to the observed loss of activity. And we also confirmed this with the functional experiment doing the cloning in the cell lines. So you can see that in the wild type BCO2, the BCO2 is disrupted. Well, it's processed, so that's you get the peak here, but then in the denaturated wild type BCO2, the BCO2 is not removed, and it's exactly the same for the mutated BCO2. They just don't the carotenoids. They don't produce apocarotenoids. And coming back to the story of the breeders selecting for the red canaries. There was a paper from two labs the same time. One was my PhD advisor just before I started my PhD, and the other from completely independent lab, looking at the genetic basis of red coloration using two completely different models. So in Miguel's lab, it was the red canary, and then comparing it with other canaries, finding two regions. And yeah, especially CYP2J19 being the one responsible for processing the red, well, to being able to process the carotenoids in order to produce the ketocarotenoids. And the other study was done on the zebra finches having the red and orange morph. And also they were able to find the CYP2J19 as the one that controlling the red coloration. So it seemed that we already knew everything. Okay, so we have the CYP2J19, so they two different species independently. But then our colleagues interested in the eye pigmentation, because I already told you that the cones that are present in the retina in birds, that they have the carotenoid pigment. So they were looking at the gene expression across red canaries. And of course, they found the gene CYP2J19, but then to double check if this is all explaining the phenotype, they again did the cell experiment. And it turned out that actually CYP2J19 was not explaining so in the chicken retina, there's the astaxanthin peak that is not present when we put the carotenoids with the construct of the cell construct with the CYP2J19. So that meant it was more genes needed to be able to produce the phenotype. And then based on biochemistry and like what kind of reaction you need to do to get to astaxanthin, they figured out that it had to be the gene related to oxidation, and then it's BDH1L. So it was a lot of molecular chemistry modeling and trying to understand what will be a good candidate. Actually, the signal in the genome, there was some, but not very strong. So that was easy to miss. And that was the case of the paper in 2016 and also another paper. And once we knew the candidate region, then you can look and then see like very, very fine signal. But yeah, without prior knowledge, that was difficult. So this basically means that from the dietary carotenoids to produce the coloration, you need BDH1L, and that's also for apocarotenoids. And for the red coloration, you have to have CYP2J19, but that's not enough, and also BDH1L. And the expression of those carotenoids can be also enhanced by TTC39B gene that I will show you in the moment. But then we were curious if that was only the case for the birds, or maybe it's similar in other species. So there was the zebrafish species that had the mutant that didn't have the red coloration. So that was a useful model to compare. And then it turns out that it's also the homologs of the CYP2J19 and BDH1L necessarily for red carotenoid coloration in fish. So it's both the birds and fish. So it seems to be very conserved mechanism across the vertebrates to be able to process the red carotenoids. And yeah, I told you already about the red and orange morph. And well, the thing that it's not in the paper, but like what we were interested, like how many genes can be behind those different morphs. So like there's a couple of different bird species that have two forms, the orange and the red. And if there's any other genes, then the one that we already know, so CYP2J19. So yeah, in 3 cases, it was exactly the same. The signal in the genome was in CYP2J19 region, so not much more to look at. But excitingly, in parrotfish, there was this differentiation in the content that they can see differently. So not having red cones affects the vision of the orange morph. And the gene responsible for this is the TTC 95B. So it's the one that I told you that enhances. So it was not. Well, so we had that signal in the genome, but then it was not really clear what does this gene do. So again, with the cell culture experiments, we figured out that when we have the CYP2J19 and BDH1L, there is the carotenoid content, but when we add the TTC95B, there was more carotenoid content. So this gene helps to enhance the signal, and apparently, if there's the mutation in it, the bird is not able to produce the red coloration. So with this, I would like to thank all my colleagues and funding from the previous work, and I'm very excited to be here. Thank you, Christian, for invitation, and thank you everyone for interacting, and I'm looking forward answering.